Hello students, in today's class we deal with Albugo. Albugo is a common fungi coming under the Albuginaceae family, and we will deal in detail of this Albugo species. Systematic position of Albugo, it comes under the class Eumycotina, subclass Mastigomycotina, class Oomycetes, order Pernosporales, family Albuginaceae and genera Albugo. Now it consists of nearly 44 species and the common disease caused by it is known as white rust or white blisters in many vascular plants, especially in Compositae, Cruciferae, then Convolvulaceae families. The members of the families, they cause severe diseases called white rust or white blisters. And some of the species of Albugo includes Albugo bliti, Albugo trapegonosus, Albugo portulaceae, Albugo candida. And the picture shows the presence of Albugo candida on crucifer leaves. And this fungi is a biotropic or obligate parasite fungus. And its mycelium is well developed, branched, aseptate, cenocytic and intracellular in nature. And one of the common feature of the mycelium is it produces button-shaped postoria. So nearly 44 species are present which mainly cause white rust or white blisters in many vascular plants. And one of the common fungi species is Albugo candida and it mainly affects the crucifer members. Dealing with asexual reproduction, the sporangiophores are formed on lower side of the epidermis and the sporangiophores bear the sporangia which are basic petal in nature. So this is the sporangiophore and it bears the sporangium which are basipetal and this is the host epidermis. So the uppermost sporangium is thick-walled and will not germinate easily and it will put the pressure on the epidermis, so the epidermis will burst open and the sporangium are liberated. The sporangia are smooth, colorless and unicellular, and based on the type of sporangia, two types of albugo are there, aeculus and annulati. Aeculus means the sporangia with an uniform thin wall. Annulati means they have some smooth as well as thick wall are present. And the sporangium varies in size from 14 to 16 or to 16 to 20 micrometer and diameter is 15 to 21 micrometer. So, during the time of asexual reproduction, the mycelium of the fungi will accumulate at the lower epidermis and will exert pressure on the epidermis during its time of formation of sporangia. Now the sporangia are borne on the sporangiophores and it exert the pressure. As a result of the host epidermis will burst open and they can be seen and they are exposed to the outer environment. Now the sporangia is mainly produced or attached in chains by gelatinous disc like tissue pad called disjuncture. So this is the sporangium. They are borne on the sporangiophore and they are arranged in basipetal manner and the sporangia are attached by means of a gelatinous pad or a disc-like structure known as the disjunctor. When this sporangia germinate directly by forming germ tube, they are called conidia. So some scientists prefer to call it as conidia and conidiophore, but majority of them support the view of sporangia and a sporangiophore. Now, sporangia detach from mycelium and spread by air or water from one place to another. At the time of germination, papilla formation take place which later form the zoospores. And the zoospores are unicellular, biflagellate and kidney shaped. After release, zoospores rust and lose their flagella and encysted, and encysted zoospore later germinate and infect to host. For the formation and germination of sporangia, optimum temperature is 10 degrees Celsius. So the sporangia are born on the sporangiophore, and this will exert pressure on the lower epidermis, and it will broken off, and the sporangia are exposed, and they are detached from the mycelium, and they spread by air or water and move to the other place. They are carried to other place, and at the time of germination, papilla formation will take place on the sporangium, and it result in the formation of zoospores. And these zoospores are unicellular, biflagellate and kidney shaped, and at the time of resting period they will withdraw their flagella and they become encysted, and later at the time of later part of the germination they will germinate by putting the germ tube, and they grow into another plant, and the suitable another fungi albugo, and at the suitable temperature for its germination is 10 degree Celsius. Then moving to sexual reproduction. Sexual reproduction is by means of oogamy. The male sex organ is known as the antheridium, and the female one is known as the oogonium. This is the oogonium, and here this is the growing part of the antheridium, and they lie side by side. Now, oogonium is spherical, terminal or intercalary and is multinucleate. Its protoplasm is divided into outer periplasm and inner ooplasm. Now, antheridium is the male sex organ. It is elongated, club-shaped and is also multinucleate. Oogonium and antheridium come in contact by a papillae which disappear shortly. At the point of contact, the oogonium cell wall becomes thin and form papillae. Through the contact of both sex organ, antheridium nucleus move towards oogonium, and later zygote form, which is called oospore. So at the time of fertilization, the antheridium will move towards the oogonium and develop papillae, and the oogonium also develop the papillae. At the point of contact that it dissolve, the male nucleus and the female nucleus will fuse together, resulting in the formation of diploid zygote or oospore. The male nucleus and female nucleus are both haploid in nature. It results in the fusion and formation of diploid zygote. So, oospore, which is the diploid zygote, are spherical, yellow to dark brown in color, and diameter is 40 to 55 micrometer, and it has a thick cell wall and is tuberculate in nature. The picture shows this one as the oogonium, and the central portion is the oospore, and this portion is the antheridium. Antheridium and oogonium will fuse, resulting in the formation of zygote. Now, before germination of the spores, they need a certain resting period. So, oospore or the zygote is otherwise known as the resting spore. After resting period, these oospore germinate by forming sessile vesicle which produce zoospores. Now, zoospores are reniform, biflagellate and uninucleate in nature. On reaching suitable host, they withdraw the flagella, get encysted and germinate by producing germ tube. And this germ tube will develop into hyphae which later form the mycelium, and a new fungi are being formed. So this is about the sexual reproduction. Now talking about the life cycle, during a sexual reproduction, the from the host cells, the sporangium will develop, and the sporangiophore will develop, on which the sporangia are formed, and the sporangia from the sporangia at the time of germination, zoospores are produced by flagellate. Zoospores undergo resting period by withdrawing the flagella. It is reniform in structure, and after that it encysted and form the encysted zoospores, and the zoospores will germinate by putting out the germ tube, and it result in the formation of hypha which develop into mycelium, and in this way a sexual reproduction will continue. Then talking about the sexual reproduction, in the hypha, male sex organ antheridium and female sex organ oogonium are formed, and both are multinucleate, and they come at the time of each nucleus will form the in the antheridium will form the male gametes, and oogonium only one will survive, other nucleus will degenerate, and the survival one will acting as the oospore or the oospore on egg, and they are surrounded by periplasm. Okay. Now the male gametes, uninucleate male gamete will fuse with the uninucleate female gamete. At the contact take place by the formation of a papilla, and at the point of contact the wall will dissolve and the male and female gamete will fuse, that is resulting in the karyogamy. The karyogamy result in the formation of diploid zygote or the oospore, and it is the diploid phase in the life cycle. Now the diploid zygote, the nucleus in the diploid zygote undergo meiosis resulting in the formation of haploid nuclei. It undergo repeated mitotic division resulting in the formation of numerous zoospores. Zoospores are reniform and biflagellate. Then it undergo the period of resting period by withdrawing the flagella, and after that they germinate by putting out the germ tube, and in this way, and this germ tube will form the hyphae which later develop into mycelium, then a new fungi is being formed. So in this way the cycle will repeat itself. So the diploid phase is noticeable by a small period which result in the formation of the zygote, which is the diploid phase, and all other stages are haploid in nature. So haploid phase is dominating and the diploid phase is denoted by only the zygote formation. So this is about the life cycle of Albugo. Thank you.